Hello friends, in this video we are going to talk about the vibrio cholerae. So it is a gram-negative salt cowed bacilli. Here you can see in this image also and fission stream appearance or darting motilities seen in the vibrio cholerae. The special media is PCBS media. From these 4 points you can diagnose uh, the case of vibrio cholerae infection. The one more point is the rice water stool. Memorize these 5 points to diagnose the vibrio cholerae infection. Gram-negative is a short-curved rod that has characteristic darting or actively motile nature. Also has polar flagella. Then it is a aerobic, which grows slightly in the range of alkaline medium. Concentration of NaCl required for vibrio cholerae is 0.5 to 1% for optimum growth, hence also called as halo-tolerant vibrio. And it can grow well in basal media, but special media is used to identify it from and also separate it from other bacteria. The media is PCBS, thiosulfate citrate bile salt agar medium is used. Is yellow colored colonies, which indicates the vibrio cholerae. Here you can see in the image that colored colonies of vibrio cholerae in the TCBS agar. There are two different types of classification of vibrio cholerae. First one is the Haber grouping and other one is the Gardner and Venkat-Raman's classification. Here we will discuss about the Gardner and Venkat-Raman's classification. So vibrio is classified into groups. First the vibrio cholerae and other vibrio species. Then based upon the somatic antigen, vibrio cholerae is further classified into 200 near about 200 serogroups, which is agglutinated by O1 antisera um, is classified as O1 serogroup, and serogroups which is not agglutinated by O1 antisera is classified as NAG vibrios. Then serogroups is classified as O1 and NAG, and in that O139 and other known O1 or O139. Okay, and then now that O1 is further O1 serogroup is further classified by biotyping in classical and Deltor. Classical biotype is responsible for the 6 pandemics worldwide, while the Deltor is responsible for the 7th pandemic of cholera. Now serotypes is Ogawa, Inaba, and Hikojima, meaning that cholera is vibrio cholerae is classified in as in serotyping as Ogawa, Inaba, and Hikojima. It can shift from one to another. The Ogawa to Inaba shift is due to the RFBT gene mutation. RFBT gene mutation. Ogawa to Inaba shifting. And one more point is that O139 is also known as the Bengal strain. So let's now talk about the pathogenesis. It is toxin mediated. Mode of transmission is due to ingestion of contaminated food or water. Infective dose is 10 raised to 8 bacilli because it is extremely acid labile. So high dose is required to bypass the gastric barrier. Uh, Factors that promote its transmission is which lowers the gastric acidity. Uh, then crossing of protective layer of mucus. Uh, it uh, once it once it reaches the small intestine, it has to cross the protective layer of mucus and reach the epithelial cells. It is mainly achieved by the because of its highly active motility. Then secretion of various enzymes such as mucinase and other proteolytic enzymes also secrete the hemagglutinin protease. It basically cleaves the mucus and fibronectin. Uh, Once it reaches the epithelium, now the next step is the adhesion to the intestinal epithelium, which is facilitated by a type 4 fimbric called as toxin coagulated villus. Now it releases the toxin, enterotoxin known as the cholera toxin. The cholera toxin has two fragments, A and B. Fragment B is the binding fragment. It will bind to the GM1 ganglioside receptors. GM1 ganglioside receptors, and the fragment A is internalized. Now, now the role of fragment A comes in. It is the active fragment, causes ADP, causes ADP ribosylation of G protein, causes ADP ribosylation of G protein, which will upregulate the activity of adenylate cyclase, results in the intracellular accumulation of cyclic AMP. The role of cyclic AMP is to accumulate the sodium chloride in the intestinal lumen. By inhibiting the absorptive sodium transport system in villus cells and activates the secretory chloride system in the crypt cells. So it basically accumulates the sodium chloride in the lumen. So now sodium chloride is accumulated in the lumen. So now what happens? To maintain the osmolality, the water also moves into the lumen, leads to the watery diarrhea. Now loss of fluid and electrolytes leads to the stroke, and also the acidosis because of also loss of the bicarbonate. The toxin of the vibrio cholerae is page coded. The genome is in for cholera toxin is CTX. There are also the other virulence factors, which includes the zona occludens toxin, which basically disrupts the tight junction between mucosal cells, and also the some of the accessory colonization factors, which helps in adhesion and the colonization. And the last one is bacterial endotoxin (LPAS). Uh, it is it has no contribution in the pathogenesis of cholera, but it is immunogenic. It is immunogenic, so is included as a component in killed vaccine used in killed vaccines. LPS of vibrio cholerae. Okay. Now coming to the clinical features. A sudden onset of painless watery diarrhea, no fever, then muscle cramps due to electrolyte imbalance, characteristic rise water stool. Complications are hypovolemic shock, muscular cramps, renal failure, pulmonary edema, cardiac arrhythmias, and paralytic ileus. Hypovolemic shock is due to dehydration, and then other complications are also there. Summarization of the laboratory diagnosis. Some of the tests we will discuss in details also. So the specimens are water stool or rectal swab, and the transport media are VR medium and Kerry Blair medium. And if you do the direct microscopy, it is a gram negative rods, so curved coma shaped, piston stream appearance. We all have all these characteristics have been discussed earlier. And hanging drop demonstrate the darting motility. Now the culture enrichment broth is the alkaline peptone water, Monsoor tellurite peptone water. Then the selective media is used is the bile salt agar, Monsoor's GTT agar and TCBS agar. TCBS in TCBS agar yellow colonies is seen. We have discussed earlier. Now the culture smear and motility testing reveals the short count gram negative bacilli and the darting motility. Now the identification is catalase and oxidase positive, uh, indole test positive, citrate may be positive and can be negative also, urease negative, triple sugar iron is so the acid gas negative, H2S negative. It is a sucrose fermenter. But remember the point is a sucrose fermenter. So into triple sugar iron test acid slash acid comes. Okay. Now string test positive. It produces hemodigestion on blood agar automated systems such as Malditov and VTEC. So the identification of the vibrio cholerae is done on the basis of these results and tests. Biotyping, serogrouping, and serotyping we have discussed earlier also. So biotyping is done to differentiate the classical and Eltor. Serogrouping is done to differentiate O1 and O139 strains, while the serotyping is done to differentiate Ogawa, Inaba, and Nikojima, which is a serotypes of serogroup O1. Antigen detection is done by the cholera dipstick assay. Molecular method is used such as multiplex PCR detecting common diarrheal pathogens, and antimicrobial susceptibility testing is also done. So now we will also discuss about some of the tests in detail. But for the MCQ or entrance examination, this table is I think sufficient to crack the question. So for the transport media, VR medium, Venkat Raman Ramakrishnan medium, then alkaline salt transport medium, and autoclaved sea water. Perry Blair medium is also used for Salmonella and Shigella. On gram staining, gram negative rods, fish in stream appearance is seen. It is curved comma shaped gram negative rods, as you seen in these images. Vibrio cholerae on blood agar sees hemodigestion. Then in the TCBS agar with yellow colored colonies, and string test is also positive for vibrio cholerae. In string test, basically when a colony of vibrio is mixed with a drop of 0.5% sodium deoxycholate on a slide, then the suspension loses its turbidity and becomes mucoid. So when you lift the suspension, as you see in the image with a loop, it forms a string. So this is the string test. For the treatment of vibrio cholerae infection, fluid replacement is done in mild to moderate fluid loss. Oral rehydration solution is given and in severe cases, intravenous fluid replacement with ringers, lactate, or normal saline should be carried out till the consciousness arrives. Therefore, thereafter replaced by the ORS. Uh, Cholera infection is mainly due to the toxin mediated, so antibiotics has a minor role. But to prevent the development of carrier stage, antibiotics can be used, and also the other uses of antibiotics are to decrease the duration and volume of fluid loss. The drug of choice is azithromycin or erythromycin for adults, children, and also in pregnancy. Alternatively, for adults, doxycycline or tetracycline or ciprofloxacin can be given areas with confirmed susceptibility. So this is all about the vibrio cholerae. I think all the points for the microbiology aspect has been covered in the vibrio cholerae, except the vaccine parts. So hope you enjoyed the video. Thanks for watching.